If you recall from the earlier mechanistic slides about transfection, it is a highly membrane active phenomenon. There's a lot of endocytic pathways involved, and there's a lot of membrane reorganization within the cell following transfection. A harsh reality, therefore, of most transfection methods is the associated cellular toxicity incurred by these techniques. Here is an image illustrating how transfection can really affect cell health. Typically, you would want your cells to look healthy and morphologically fine, have no effects on cell viability, etc. following transfection. These are HeLa cells that have been cultured and transfected using several different transfection reagents. A lot of different labs use HeLa cells as their cellular model. Unfortunately, oftentimes, depending on the transfection reagent that you used, your cells will end up looking like this. Now let's look closely at these HeLa cells. Depending on what transfection reagent you use to transfect these cells, here are a few examples. The first on the left is our Transit LT1 reagent, followed by Transit 2020, another high-performance reagent by Mirus. And in comparison is a liposomal formulation that is popularly used called lipofectamine 2000. At the 8-hour time point, you barely notice any differences in cell morphology with either of these reagents. The cells look just fine. However, as you progress into the 24-hour time point post-transfection, the lipofectamine 2000 formulation wreaks havoc on the cells. The cells are all shriveled up and rounded compared to the healthy-looking cells with the Transit LT1 and Transit 2020 formulations. This also correlates with the GFP expression profile because the cells that are rounded up and shriveled, even though they might be expressing some GFP, are essentially lost during transfection. This drives home the point that striking the balance between expression and toxicity is really critical for your transfection experiments. Apart from just visually assessing cell health, there are a lot of very sensitive assays available to detect cellular toxicity. One that we regularly employ in our labs is the lactate dehydrogenase assay, or LDH assay. Lactate dehydrogenase is an enzyme that is expressed by all cell types. What this assay does is assesses the membrane leakiness by assaying the amount of LDH release into the medium from the cells. So indirectly, it assesses the level of membrane damage because you have an otherwise cytosolically located enzyme that is now getting released into the supernatant because the cells are leaky. Here you can see a data set of HeLa cells that have been transfected with a luciferase encoding plasmid. This data shows expression and also the corresponding toxicity values in terms of the LDH release from the matching cell supernatants. Again, as you can see here, with lipopoliplexic formulations such as Transit LT1 and Transit 2020, you see good expression in these HeLa cells and correspondingly low toxicity. However, with liposomal formulations such as lipofectamine 2000, the expression of luciferase is twofold lower, at least, and also the levels of toxicity are very high, close to 60% compared to less than 10% with the LT1 and 2020 formulations. With these gross effects on morphology, viability, and membrane leakiness, one wonders what really happens inside the cell. What causes the cells to look so sick or vice versa? We looked into this further by looking at the gene expression profiles by using an RT-PCR-based method to analyze the expression of 84 key genes involved in cytotoxicity. This experiment was done with HeLa cells that was transfected using either Transit LT1, Transit 2020, or lipofectamine 2000. In this experiment, a non-coding plasmid was used to make sure that there were no gene expression associated changes on the overall cytotoxicity profile. As you can see here, cells hugely alter their gene expression profile following transfection. Of the 84 key genes that were looked at, the alteration in gene expression was maximum with liposomal formulations such as lipofectamine 2000. Expression of more than 9 and more than 25 transcripts were altered by lipofectamine 2000 transfection at 8 and 24 hours respectively. Compared to this, very few genes, only 3 and 9 respectively at the 8-hour and 24-hour time point, were affected by cells that were transfected using the Transit LT1 and Transit 2020 complexes. You can get a further idea of the magnitude of the gene expression alteration by looking at these results in the form of a Venn diagram. As you can see here, many more genes are upregulated or deregulated with lipofectamine 2000 at either the 8-hour time point or the 24-hour time point. Could these gene expression changes be affecting the pathways that you're studying? We conducted pathway analysis using the Ingenuity software on these RT-PCR results, and we found that the most overrepresentative pathways that were affected by transfection were associated with oxidative stress response and xenobiotic metabolism. Among the genes involved in oxidative stress response, that is shown here in the topmost panel, the DDIT3 mRNA expression was increased approximately 50-fold in cells that were transfected with lipofectamine 2000, whereas it was significantly lower with cells that have been transfected with either Transit LT1 or Transit 2020. Based on this pathway analysis, we also found that the most significant biological function affected in response to transfecting cells with lipofectamine 2000 is related to unfold protein and DNA damage response. This pathway includes deregulated genes coding for heat shock proteins such as HSPA5, HSPA1A, DNAJ, subfamily B, CDKN1A, and tumor protein 53. What was concerning to us and could be to you as well is that a lot of people are actually doing the research on these particular pathways. So if you use transfection reagents that could significantly upregulate or deregulate the gene expression profiles of any of these proteins that you're looking at, it would be a major concern for your experimental output. Furthermore, it is seen that different cell types show different expression toxicity profiles than, say, HeLa cells. Here are a few examples of HepG2 cells, THP1 cells, and D2 cell and HUVEC cells. As you can see here, you do get a good balance of expression versus toxicity that is measured here in terms of luciferase expression versus LDH release with either Transit LT1 or Transit 2020 in most cases. With lipofectamine 2000, you can get moderate levels of expression, but the toxicity levels as measured via the LDH assays are exceedingly high. So this further emphasizes the point that it is very critical to choose the right transfection reagent for your particular cell type. You can also learn more on our website www.thetransfectionexperts.com. We have a technical resources section that you can find useful for gaining more information about transfection. We have tips from the bench there to help with particular experiment types such as DNA transfection or siRNA transfection. We also have a comprehensive transfection database called the Reagent Agent, which gives you recommendations for your particular cell type and nucleic acid combinations. You can also speak with the transfection experts directly by either calling us or emailing us at our tech support address. Thank you so much for watching and listening to this tutorial. If you have any feedback to give to us or any questions, please do contact us.